All right, Chapter 8 is going to talk all about genetics. Some of this information you may be slightly familiar with because molecules such as DNA, RNA, proteins, we've already talked about those in regards to them being part of those macromolecules. And then depending on what your background is, some of these terms might be slightly familiar or even very familiar with you. And then some of the material you may have actually heard to some degree, whether in detail or just big picture, in the just general media, all right, in the news and just other science courses that you may have had. So for section 1 and 2, we'll see if we can get through both of those sections in this video component. You've got a variety of terms in Objective 1, and then really what these two first sections talk about are DNA and protein, how you go from one to the other, and then the processes of transcription and translation. So Objective 2 is how DNA, RNA, and protein fit together in terms of the two processes of transcription and translation. In addition to the PowerPoint notes, you also have a couple of videos to watch, and that's going to give you a lot of additional information and a repeat of some of the very detailed information that you have in your textbook. But just big picture, when we start off with section 8.1, introducing genetics and genes, you have two different key terms of genotype and phenotype. Genotype is going to be the sum or the genome is the sum of all of your genetic material. But genotype is going to be specifically to say what gene, what is the DNA that you have that is going to code for a specific trait. And then phenotype is going to be the physical or outward expression of the genotype. So what color are your eyes? Green. All right, so if your eyes are green, then green eyes is your phenotype. But on the interior, you have specific genes. Your DNA has the gene that's going to code for and result in you having green eyes. So the DNA component is your genotype, and the phenotype is your physical characteristic. All right, and so what you see on the lower part of this slide is a phenotype. All right, do you have unattached earlobes or do you have attached earlobes? And that's just the very bottom of your earlobe. And a genotype would be two big E's because that gene, the big E, is going to be a dominant gene. And if you have both dominant genes, then you're going to have the dominant trait, which is the unattached earlobe. And then if you have one gene, a dominant gene from, say, your mom, and a recessive gene from your dad, your body is going to express the dominant trait. And so you would see the phenotype of the unattached earlobe, although your genotype is big E, little e. And then if your genotype are the two recessive genes, the two little e's there, then you would have attached earlobes. And so there you can kind of get the distinction of genotype and phenotype. All right, so the core ideas of looking at gene expression. How is the material that is encoded in your DNA expressed to give you the physical traits that you have? Well, we begin with DNA. All right, the DNA is going to be in your chromosome or found within your chromosomes. Your chromosomes are the DNA. These are long-lasting. You find your DNA in the nucleus. And then you have your RNA, which is going to be transcribed from the DNA. RNA is going to be small molecules. It's short-term because it actually gets degraded after it's made, shortly after it's made. And the information is going to be used, the RNA is information used to transfer from the nucleus to the entire cell. And that is what is going to occur in eukaryotic cells or in your cells. All right, and the RNA is going to act as the map, so to speak, to get your protein. Proteins are going to be different sizes, they're going to be expressed at different times, but the protein is what carries out the genetic function. Alright, so hopefully you remember just the core features of DNA. Alright, a couple of different ways to look at it here. Alright, in part A, you have the double helix. DNA is double-stranded, and so you can see that a little better here where you have the backbone of the DNA, and you can kind of think about this as the DNA ladder, which is sometimes referred to. You have the backbone, which are the phosphate groups that you see here, the sugar, and this is DNA, so it's deoxyribose. And then you have the nucleotide, the nitrogenous bases. Alright, so you have thymine, you have adenine, which pair up together. You have guanine and cytosine, which pair up together. And these create basically the rungs of the ladder, the steps, and they are connected by these hydrogen bonds. So hydrogen bonds are actually relatively weak, but you have a lot of these bonds, and so therefore it's actually a pretty strong connection. And then your double-stranded DNA molecule is going to be a helix. It's going to twist, and that's what you see here. So, overview. DNA is a double-stranded helix, like I mentioned, that you see on that previous slide. It is composed of repeating nucleotides. All right, there's your three components of DNA. The sugar and the phosphate make up the backbone, while the bases make up the rungs of the ladder. And you have that complementary pairing, cytosine with guanine, adenine with thymine. Now, another big idea within DNA is going to be DNA replication. And this is the topics here that we're seeing in just a few slides is actually an entire course. All right, you can have a genetics course where you're going to take 15 or 16 weeks to talk about this material. So what you're getting here is really just a big picture of what's going on. And if you understand the big picture, then you're going to do well with the information that you need from here on out. All right, so here's your DNA helix up here, and the DNA needs to be replicated if your cells are going to divide. And so you have the parent strand of DNA. It's going to be equivalent over here. All right, and then enzymes are going to come in and break that double-strand bond here. And so then what happens is you have new DNA molecules that can be created, it can be replicated by material coming in and filling in. All right, so this yellow strand here is the new strand, and the old strand here, and then over here is the old strand. Those separate, and then new nucleotide material comes in and fills in this part. All right, so the bluish-green color here, this is the parent strand, and the purple side is the daughter strand. But now you have two identical strands down here, as with the parent strand, because DNA replication has occurred. All right, now a quick review on RNA structure. Very similar to DNA, but it's only one strand, so it's single-stranded. And then you also substitute out the thymine for uracil. You still have guanine, adenine, cytosine, but uracil replaces the thymine. All right, the RNA is actually going to be the chemical language that can be read by your cell. So therefore, even though you have DNA in your cell, the cell doesn't know exactly what to do with that in terms of overall function. So you have to have an mRNA sequence, or that RNA sequence created or transcribed, so that your cell can have something to read in order to facilitate the regular processes within the cell. So 2 steps in gene expression. You have to make the message and you have to read it. So the 2 big ideas here, the 2 big processes, are transcription and translation. Transcription is DNA is read to make an mRNA sequence in the nucleus. Again, this is a little easier to think about for the eukaryotic cells in terms of this occurring in the nucleus. So that's why I'm using this material. So you have the DNA helix. The DNA is going to be transcribed to create an mRNA sequence, which you see down here. All right, you notice this is mRNA because on this bottom strand you have the U instead of the thymine. So the U binds to the adenine. The adenine still is in the RNA sequence, but that binds to the thymine. All right, here's another uracil binding to the adenine, and this is just the overall sequence that is then going to be used in translation. Translation is reading the mRNA to make a protein, and this occurs in the cytoplasm, and it's going to be facilitated through a ribosome. All right, so the overall processes of transcription and translation occur in two different areas of the cell. This figure is not in your textbook, but I just thought it was a good one to get the idea of what's going on. You have your nucleus here, where you have your DNA, and that is created or used to create an mRNA sequence, which you see here. The mRNA sequence is moved out of the nucleus through a nuclear pore into the cytoplasm, and it's here that a ribosome is going to come and attach to the mRNA sequence, and that's going to facilitate the creation of this peptide. All right, individual amino acids coming together to create a protein. So this is figure 8.5 from your textbook, and it's pretty basic. It's just showing the big picture ideas where you have your DNA. The DNA is transcribed to give you a variety of different mRNA molecules. You have transfer RNA and mRNA and ribosomal RNA. And the mRNA, the M stands for messenger. So then, this is in the nucleus. This occurs, is created in the nucleus, leaves the nucleus and goes to the cytoplasm. And then you have translation of the RNA. Translation takes the mRNA sequence that you have down here, this little strand, in connection to the ribosomes. And ribosomes are made from small proteins and these ribosomal RNA molecules. The ribosomal RNAs are very, very small fragments of RNA. Those two create the ribosome, and then you have these transfer RNAs. And the transfer RNAs are going to have specific codes here that are going to attach to the mRNA sequence, and that is going to dictate what amino acids come in and are added to this chain of amino acids. And then the chain of amino acids is what's going to be your protein. And so at this final phase down here, you have expression of the DNA through a protein for structures and function of the cell. All right, and that is it for sections 8.1 and 8.2. Again, watching the other videos is going to give you a little bit more detail and going to add to that understanding of the big picture.